Good morning, everybody. Thanks to the nice introduction to Christian. I would like to thank the organizers to inviting me here, and I hope you will have a good day, and it's a pleasure for me to open the session. Okay, so what I would like to address in my talk is how can phenotypic screening or assays or approaches improve the success of drug discovery. And you might have seen the analysis of Swiney and Anthony some years ago when they published the study what approaches led to the most first-in-class drugs. There was the publication in the Nature Reviews Drug Discovery. And they found out that more phenotypic approaches were leading to first-in-class medicine. And quite recently, there was a publication, so it was in the August issue from Nature Drug Discovery, so that there were two analyses being done from two different groups. So one actually from our groups, from our colleagues at Novartis, and the other one were more focusing on cancer drugs. Where did these first-in-class medicine work come from? So which approaches led to that? And now there was a different view, and that was based first on the time frame which were analyzed. There were the later years also being included, the drugs from until 2013. But also they distinguished a little bit more what are really phenotypic approaches, because you can, it's a wide field. You can also have mechanism-informed phenotypic screens, or you could have pathway screens in there, and there's also a bit of rationale of the mode of action behind. So it's a big discussion, and it's a big discussion especially there because there is a high attrition rate in the drugs, and there is a lack of targets. So coming to phenotypic screens, we in industry, we hope that this would lead to new drugs without maybe knowing the target, so it could be agnostic of the target. And you could also try to identify the target later on if necessary. There will be an increase of complexity with phenotypic screens, but hopefully also more physiological relevance. And we recently have seen that there, with the technology improvement of high content assays being able to be done in a higher throughput, and very recently also the kinetic live cell analysis assays, we can set up phenotypic assays quite early in the development or in the research. Sometimes as primary screen, but also early as secondary assays. With that, we did an analysis of the approaches. So here you have to be careful, it's not the drugs which came out, so it's not the same analysis as the others, but it's rather the approaches, so the assays. How many assays were done with target-based versus phenotypic approaches during the last years? And this is now an analysis for the Novartis Institute of Biomedical Research, so for the NIBR within Basel and Cambridge. The data, and here we saw really an increase over the last years going phenotypic, and that went along with the development of the technologies, as I said, for high content readouts, and very recently for the live cell imaging, which I will show you later as an example as well. So this is just an overview of the screening facilities which we have for the specific case of cell-based assays, phenotypic assays. We can do that in high throughput. We can do that in 1536, if possible, from the setup of the assays. We developed, we engineered the automation that we can do washing in 1536 and immunostaining and so on. So the technology is there, so now we have to see if the assays can be set up in that format. And this is just a short overview of our imaging capabilities, like the Opera, we have InCell 2000, and the Yokogawa CV7000, which can do live cell imaging in a quite automated fashion. All right. With that, this was the, how we can do that. Now, if we would do phenotypic assays later on, how do we, develop further? And often the question is, can we derive the target, and how would we do that? So there are different possibilities, which, always depend very much on the projects, and it very much depends also on the sensitivity of either the compounds, what you have, or if there are available mutants for the specific targets. So you can do chemoproteomics approach or affinity chromatography, but if your compound isn't potent enough at that time point to pull down the target, you might fail in that setup or in that approach. We also exploited, because of some difficulties with the compounds which we derived from phenotypic assays, we exploited the possibility of target hypothesis generation by in silico means. And this is what I would like to explain a little bit more. So we generate fingerprints by high content imaging of targets which of compounds where we know the mode of action, and then try to bring that back or to cluster that together with compounds with unknown mode of action. And the idea is here that you would have a fingerprint or a phenotype from a compound where you know the target, and if you don't know the target of that compound, you could then generate their target hypothesis through the same phenotype. And they might also have different targets, but might be linked in the pathway space. So that is the hypothesis behind. That's what we tried with the setup with 2,700 compounds, which were well annotated. And we generated readouts through high content screening, so cellular phenotypes with fixed, with a fixed setup with immunostaining. We generated as many features as possible, and then combined that into an HCS fingerprint. And you would see here, this is, do I have a pointer actually? Now I'll do it like that. So you have the neutral control would be here, that black one, that's the DMSO control. And then a positive control where we know the mode of action of the compound would score like that one. These are all the readouts which we have. And then you could assume that an active sample with a similar phenotype would have a similar target or being involved in the same pathway space. And we did that with 6 different readouts across these compounds. We screened these. We took these readouts for different compartments. Like you can see here, for, of course, the nucleus, we took the Höchst. Then the cytoplasm, we wanted to identify with the cell mask stain, the deep red stain. Tubulin with an anti-tubulin antibody, and the ER, we took the anti-K-del antibody, the Golgi compartment with the anti-giantin, and mitochondria, the anti-TOM20 antibody. And we tested if compounds where we know they would interact or would generate a phenotype on these compartments. And here you see some of these tool compounds like topotecan, nocorazole, and especially here the chloroquine, you see nice vacuoles in the cytoplasm. And here for the mitochondrial membrane potential uncoupler, we see a dispersion of the mitochondria, for example. So with this in mind, we then set up and went to the whole 2,700 compound testing, generated these fingerprints, and pooled them all together because they were done in separate experiments, and generated a fingerprint. There were some highly correlated features, so these were removed, and in the end we generated a fingerprint of 86 dimensions. The analysis was done like very often with multi-parametric data. You take the statistics from, you use the calculation of the Mahalanobis distance. And what we found out, I mean, these were highly active compounds in general, but over 50% were active. And we defined active being, different than the DMSO control, more than 5 MET. So the medium activity distance, active distance. And then we wanted to, so we had some annotations for that, but we wanted to cluster them together and then do the gene enrichment set analysis with data from external databases. And we did that by the MCL clustering mechanism. And here we saw that we resulted in 11 or 12 resulting, in 84 resulting clusters, which we'll show you in a second. But we saw one point which I want to mention here is that with the number of the measured cellular compartments, so meaning you had all of these readouts being taken, so together you get the more active compounds. Because in the end, in the beginning, we were not sure if we would need all the 7 or 6 compartment stains, if we could, spare 1 or 2. But we really saw with increasing with increasing staining of the compartments or stains, we saw an increase of the active compounds, and they were not redundant. Okay, so the MCL analysis resulted in these clusters. We had highly enriched or significantly enriched clusters, which you can see here, as well as with the quadrants, and the others which are enriched as well, but not significantly. But I want to mention this nevertheless because there were maybe not too many compounds in there. That's why it was not significant. But it was clear that there were 3 out of 4 being annotated with a specific target. So the analysis has shown that quite a lot of them were enriched for kinases. You see that in the gray shaded clusters here. So with a gray shading behind. Our library is enriched for kinases. That is maybe shown also in that setup. But there were also quite a lot of other distinct targets being identified. And some of these clusters I want to highlight a bit more. So, for example, this one, which is here, and this is based on a minimum spanning tree. This is based on the distances of the mediate of the HCS fingerprints. So they are correlated to each other. And as you could see with the kinases. And here these 2, which are very close to each other, were both very highly enriched for HDAC inhibitors. You can see also so here is the fingerprint, meaning, so we have here all the nuclear readouts, here the cytoplasm readouts, tubulin, and so on. And as you can see, this is the overlay of all of these compounds in that cluster. They are pretty well, or pretty similar, as well as with the cluster 17. They have some differences, that's why they're not clustered together. But you can also see from the image that they are pretty have the same shape. They're elongated. There are less cell number, of course, in there, and they were all annotated for HDAC inhibitors. The other cluster, which you can see up here, is the cluster which is highly enriched for PI3 kinase and mTOR inhibitors. And the interesting thing here is that you can see in the cytoplasm there's quite some activity or some special signature. And I hope you can see that, because here we have quite nice vacuoles being in that cytoplasm, and as well as in these other cluster 9. This one maybe you might see, this is the strongest over there. And this, of course, can be within the autophagy or autophagosome formation that PI3 kinase inhibitors lead to this vacuole formation. So with these HCS fingerprints, if we cluster now compounds with unknown mode of action to this, we could set up target hypothesis of these compounds. One last example is about the tubulin inhibitors, where we found that the tubulin readout wasn't the most striking one. You see a loss of the tubulin network in here very clearly. However, we saw a very strong signature for the Golgi. So we checked a bit more in the Golgi staining channel and saw a dispersion of the Golgi stacks. And it has been published that with a microtubule inhibition or dispersion, also the Golgi stacks are affected, and you see that pattern that was published 1999 already from a group. So with this, we're pretty confident then to link the compounds to that. That's what we try. Currently, we set that up. Of course, so we tested it with one concentration with 10 micromolar, and you could argue about off-target effects. So we did that also with lower concentration and in dose response. So we are currently analyzing that data. The second point is the cell lines. We should go in the relevant cell line of the project and do that analysis. That's what we're going to do with project compounds from phenotypic screens. With the markers, you could still argue you need other markers, but we were pretty, satisfied with this, with our choice. So these were now the phenotypes from cells, from fixed cells, to generate target hypotheses. Phenotypic screens, what we would like to use in more, increasingly, are hopefully more relevant if you use live cell. So for one project, we wanted to set up a very physiological assay to mimic the NMJ. So for a muscle disease targeted project, we wanted to set up the NMJ via a co-culture. And you know that for either sarcopenia or ALS, the amyotrophic lateral sclerosis, we see a denervation of the axons, which are here stained in green, from the muscle cells, so from the muscle junctions. And we were hoping that by mimicking the NMJ in a dish, we could try to either, increase the innervation or find out what mechanisms are behind which really retract or denervate the neurons from the muscles. So what we set up in 96 well plates recently is that phenotypic assay with differentiated human skeletal myotubes and red spinal cord extracts or motor neurons from red spinal cord, so from embryonic red spinal cord. And we incubate that for quite some time. And here you see 8 days of co-culture of the two cells. And now I hope you can see that. You see the muscles contracting here. And this is being imaged live with a video over, here we took 60 seconds, 2 seconds per frame, so 120 images in a bright field mode. And now we were, wondering how could we analyze that quantitatively to compare a compound screening, in a compound screening mode, one well to the other, one setup. So the first thing what we tried, or the first analysis what we tried, was this time series analysis, where you would record or compare a pixel in one image, so one pixel to the other in the other image, and really at that pixel part, so really the same location from one image to the other. And there's these publications about that it's best to do that via a window size of maybe 5 or so. We evaluated that as well. So we recorded that and came to that analysis where you would see now here, these are the 9 fields. So we're recording 9 fields in a well. So this is one well. And you see for the one field, for example, there's no contraction at all. In a 96 well plate, you need to record several fields to have quite a good covering of the well. And then for another one, you see quite a lot of contractility in here, which is maybe not very, these are similar to this one, so it could be the same, but that looks different, or these look very different from each other, so are these different contractions in there? We're wondering. you can compare very nicely on the left-hand side an active well versus on the right-hand side an inactive well. However, we realized that with this method we have quite a lot of background issues, because here we are analyzing the intensity, so the pixel intensity from one location or from a specific location to the same location in the next image. So we're not tracking the pixel itself, but rather looking if there's an intensity change. So you can nicely visualize, how does that work now? Visualize the changes here. This is the analysis, and that's how we then compared if we are doing the right thing. And then realize that small debris, for example, would give us a signal, or that it is very much dependent on the contrast of the recording. And this will always vary from well to well. So it was not very stable or not very robust, that method. So our IT colleagues really did a nice job in identifying the optic flow computational measurement and set that up into a little program, software program. So here you're really following the pixel itself. So not comparing always the same location, but really following the pixel, and that could be also being used for cell tracking. You don't want to follow really cell tracking like in cell migration, but you could use that as well. And here you get two informations. So first the magnitude, but also the direction. And this is exemplified here with this vector. So these pixels are really followed over the time, and then you can more clearly say what is happening and what is the movement of that specific pixel. And how does that work? So finally, you have to do some binary setup that you really get that into a quantification. And what you can do then is to have the same, illustration. But now you see a difference between when does it really, you could see that as well, the difference in the space. But here, based on the color, you can see where is it going in which direction. And this we can use for really, identifying which contractions are at the same time, happening at the same time or later, and that is important for a later analysis as well. So here, you might wonder, so how do we get now a value out of it to do compound screening? Because that's, in the end, it's a nice method, you see that nicely, but how do I now compare compound X versus Y, or concentrations, or over the days of co-culture? The trick in that sense is to sum it all up to get a kind of an area. And here you see that what is really projected from the three-dimensional space into the two dimensions. You have that area coverage, so you could get this as a value. You say now a contraction, you saw small contractions before, and we could compare and say if now after 10 days or 12 days in co-culture, it's much more contracting versus 7 days of co-culture. That could be one value to distinguish. We could also use this information to get different, different contractions. So if we have now neurons innervating the muscles and we want to increase the number of innervations, or compare that versus a patient and say in a patient cell from an ALS patient we have less innervation versus a healthy donor, then it might be important to see, can we increase the number of contractions in a well. So for this it is important to distinguish between the different contractions. And we can do that by analyzing here the time points from the binary data and group them together. So you clearly see these all have the same pattern, and that is really done over the whole pattern, and then group them together and say this is one contracting unit versus another contracting unit. And finally, we put that into a segmentation using CellProfiler to identify then also the area of the contractions and the number of contracting units. The frequency we could do as well. We are working on that. That's still a bit difficult, but that's what we are setting up currently. And the analysis is then being done like, you could say, you compare the area of the total area of the contracting units, or you could take the number of contracting units. So here, for example, we have 5 contracting units in there, depending on the coloring, versus, you just have 1 in here. And here it's the same field, but we want to compare different conditions. And we did that. We compared quite a lot of conditions, so of the days in co-culture. The myotube density is very dependent, or it has a big influence how many human myotubes are co-cultured. Then the plate coating with collagen, laminin, and so on is a factor. And we also saw a difference between if you use plastic or glass plates, for example. It makes a difference in the contractions. And that's what you can see here, that we have the days in co-culture. We have an increase over the time, but also after a certain time, then a decrease. So then, although they always get a new medium, they're always done with medium change, you see a drop of the contractility as well. So we are currently setting that up for, with that quantification, which we realized here, we're setting that up to do a phenotypic screen, which really resembles the neuromuscular junction. We currently do that with red spinal cord extract, but we are in the development of using iPS-derived cells from patient, and we'll compare that with healthy donor cells. But also with the rat, we try to get muscle also then from sarcopenic rats to improve with the compound the effect which we see in sarcopenia. And this is what we think would really help in the drug discovery. And no matter if it's target-based or coming from a phenotypic approach, I think really that it is important to have the clinical relevance in there. And with this setup, we hope we are very close to the physiological relevance to the disease, especially for if we use ALS patient cells or then cells from sarcopenic rat. To test our compounds early on in that. If it's a primary or a secondary assay, it doesn't matter so much, but really to have always the clinical manifestation in mind. And I think Sam Stott made that statement nicely in one of the publications. So with this, we hope we get nice and important drugs soon. The sooner the better, I would think. And finally, I would like to thank everybody who was involved, quite some people involved. So Felix Reisen was our postdoc, and Amelie, our student. They did the fingerprinting work together with Paul and Shan. We did the data analysis, and lots of people were involved for the discussion. And on the right side here, you see Imtiaz and Gianluca, who are very, very important. They developed the optic flow method and the whole setup for the image analysis of the live cell imaging. And these are all the project teams from the MSD project. And with that, I would like to thank for your attention, and I'm ready for questions.